Hello, I'm Mr. Wilkinson. Today we're going to be talking about the different kinds of cells. If you'll have a piece of paper out and something to write with, you can follow along with me as I draw. So when we draw, it'll help us remember what we're doing. So let's start with the title of today. We're going to be talking about cells. Specifically, we're going to talk about two kinds of cells: prokaryotes and eukaryotes. So let's start by breaking these words down. We'll start with this prefix pro, which comes from proto, and it means before. This prefix eu means true or good. And then let's link it with the root word that we've been using, karyote, which means nutshell, which is referring to the nucleus. Okay? So what this means here is that prokaryotes have no nucleus. They came before the nucleus. And eukaryotes have a true nucleus. So we can just kind of break these two groups down. All right, so prokaryotes came before the nucleus, and eukaryotes have a nucleus. Throughout this whole discussion, this is our central thing we're going to keep coming back to, that prokaryotes don't have a nucleus and eukaryotes do. All of the other parts of these two cells come down to this one fact. So let's start by just talking specifically about prokaryotes. Prokaryotes. They came first. They're simpler than eukaryotes. And I think really to help us out, we should just draw them. So don't worry about your drawing. It doesn't have to be awesome. Just draw along with me. We're going to take it simple and slow. We'll start with a little oval like that, okay? This is the cell membrane. If you remember when we talked about the characteristics of life, all living things have a cell membrane, and these membranes, right, define where the cell is and where it ends, where it begins and ends. Just outside of the cell membrane is the cell wall. So draw another line going around your oval, just like that. Okay, this is the cell wall. We'll go ahead and label them really quickly. We've got our cell membrane, which defines where the cell is. And just outside of the cell membrane is the cell wall. This cell wall says where the cell begins and ends. On the inside of the prokaryote, there are these little structures that are very important for all cells, and these structures are called ribosomes. Ribosomes. Okay? And then in the middle of the cell, these prokaryotes have DNA. This DNA makes a ring structure, but it's all kind of looped together. Like imagine having a really long piece of yarn, and then you tie up the ends, and then you kind of wad it all together. That's what's going on here in the middle. So you can make just kind of a bunch of squigglies all together in the middle and then try to connect them together. And that is the DNA for prokaryotes. And it's this in this area, which gets a special name called the nucleoid region. The nucleoid region is this middle of the cell where we have all of this DNA all gathered up. That's all you need for a prokaryote. But some prokaryotes have another part called the flagella. It's this tail-like structure that comes out of the cell. Okay, this would be the flagella. Flagella. All right, now we've drawn our prokaryote. Let's talk about the different parts of the prokaryote and what they do. So the cell membrane is very important. It regulates what goes in and out of the cell. It can decide which molecules to bring in and which molecules to push out. So just like how you decide what goes in your house or in your room and what goes out, right? You're bringing in food into your house and you're pushing out waste from your house. That's exactly what the cell is going to be doing. And that's the main job of the cell membrane, to regulate what goes in and what goes out. Just outside of that, there is the cell wall. The cell wall of plants and animals animals don't have cell walls. Bacteria, right? These cell walls keep them from exploding, okay? So for these prokaryotes, they have this special rigid structure on the outside that provides extra support for the cell. So we'll talk later about exactly how that happens. But essentially, if too much water goes inside, they can bust open like a water balloon. So these cell membranes surround them, right? The cell wall surrounds the cell membrane to keep them from exploding. You can think about a cereal box and how you can have like that bag in the inside of the cereal box, and it goes inside, and then the box around it provides extra structure for that bag. That's essentially what the cell wall is doing. This next one, ribosomes, I want you to star this one. Put a little asterisk, a little star by your ribosomes. They're very, very important because they make proteins. They make proteins. Proteins allow the cell to have a metabolism. They are the tools of the cell. So if we don't have ribosomes, we don't have the tools we need. We don't have proteins to have a metabolism, and the cell cannot function. So notice how these different cell parts are correlating to the different functions needed to have a living organism. Right? This cell membrane here is working with homeostasis, bringing things in and out. These ribosomes are working with making proteins, which is directly related to the metabolism of the organism, what they can do with breaking food down or building new parts of the cell, all happens with these proteins. Next, we have the nucleoid region. The nucleoid region is also important because it has the DNA. DNA is like a library for instructions to make proteins. So these ribosomes are working really closely with the cell's DNA because they're using that to make the proteins. Now this DNA also is going to be how cells pass information down from one generation to the next. So like how your parents gave you information, these bacteria are going to give information to their offspring. So you can eat food, all kinds of food, and you can do that because of the proteins you have. And bacteria, these prokaryotes, are going to be able to break things down based on the DNA they have. And when these bacteria divide and they reproduce, they're going to give all their DNA to their daughter cells. And those daughter cells then are going to be able to do everything the parent cell could do. So this DNA is the heredity. That was another characteristic of life, right? Being able to pass traits down. And over time, they're going to be evolving because this DNA is going to change just a little bit every single generation. Okay? Then flagella. Flagella allow for movement. And so, because flagella can move, it means they can respond to their environment. Now, not all bacteria have flagella, and they don't all need to have flagella in order to respond to their environment. But those kind of responses happen more at the biochemical level and are a little bit more difficult to explain here for our introduction. But I want you to understand prokaryotes, even though they're just one cell, they can do all of the things that living things need to do. They can maintain homeostasis, they can have a metabolism, they can have complex heredity, they can pass their heredity down, they can grow and divide, they can evolve over time, and they can respond to their environment. They can do all of those things. Every single prokaryote can do this. Now let's take a look at some common shapes of prokaryotes. So I'm going to go to a new page. I'm going to set that aside. We're still talking about prokaryotes. If you still have space on your page, you can continue drawing and writing on your page, but I'm just continuing. So there are 3 common shapes of bacteria I would like you to know. There's one that's kind of oval or rod shaped. There's one that's spherical or round shaped. And there's one that's like spiral shaped. Okay, and we're going to give names to all of these. There's special scientific names that we give them so we can sound smart. Okay, that's what we're going to do. So this is a rod shaped or a pill shaped. That's the common name, so we're going to put that in brackets. That's not what we're really going to call it. We're really going to call it, so that we can sound smart, is bacilli. Bacilli. Or if we're doing singular, bacillus. So this would be bacillus, and if we had a bunch of them, then they would be bacilli. All right, that's the pill or rod shaped. Next, let's talk about the spherical shaped. Spherical or round shaped. These are called cocci, or if we just had one, coccus. Okay, it just means spherical, it's a ball. Last, we have the spiral shaped. Spiral shaped. And this one I think is easy to remember because it kind of sounds like spiral. They're called spirilli. Or singular, spirillus. So hopefully you've seen this pattern, right? Because these are Latin roots, these are Latin words, we're using I for plural and us for singular. And all of these words do that. Okay, so these are the 3 common shapes of bacteria. Bacteria can take other shapes as well. And, you know, I've been saying bacteria a lot, but bacteria and prokaryotes, they're the same thing. We'll talk about that in a second. So prokaryotes, they have these 3 common shapes. And there are other shapes that they can take, but you just need to know these 3. Okay, make sure you know bacilli and cocci and spirilli. All right, now I said that there are different kinds of prokaryotes. We talked about bacteria. Maybe you've heard this around. They say, you know, they got a bacterial infection, or wash your hands, clean off the bacteria, something like that. In science, bacteria are sometimes called eubacteria. And if you'll remember, this prefix eu means good or true. So they're calling them eubacteria to distinguish them or separate them from this other group called archaea. Archaea, this word comes from archaic, and it means old. Okay? Archaea, old. All right? So if we look at bacteria and archaea, they look very similar. They're going to be similar shapes. They seem to be doing the same kinds of things. But if we look at their DNA, if we look at the information inside of their cells, these archaea actually look more like eukaryotes than they look like these eubacteria. So that's why we give them their special name of eubacteria over here to distinguish them from these archaea, right? They used to be called archaebacteria, but we no longer use that term. right? And we've called them eubacteria now to kind of distinguish them more, to separate them, because archaea look a lot different genetically. When we look at their DNA, it's very different than bacteria, okay? Now, another thing that archaea have is that they're sometimes called extremophiles. And they're called extremophiles because they live in really harsh conditions. They can live in very salty areas like the Dead Sea. It's not actually dead. There's archaea that live there. They can live in really, really hot areas, right? Like hydrothermal vents at the bottom of the ocean that are boiling hot temperatures. Archaea can live there. They can live in really cold areas, and some of them even live inside your guts. So these archaea live in very extreme places, so they sometimes are called extremophiles, but that's more a descriptive term and not a name for them, if you will. All right, so that's the two groups of prokaryotes. Now, if you remember, these organisms do not have a nucleus, okay? Now, I want to go back to this. Archaea, we think, look most like early life today. So I want us to think back about what early life might have looked like. Today's cells are incredibly complex. Even these simple archaea are very complex to what an early first cell would have looked like. These first cells all would need just some way to have a very simple metabolism, copy itself, and divide. And then over time, as these cells would divide, they would have gained new traits and abilities. And with every new trait and every new ability, they would be able to survive even better than the generation before. And those things would stick around. So keep that in your mind. This early life also lived without oxygen. Oxygen is pretty reactive, and it would want to bind with different things in the environment. And so there would not be a lot of free oxygen floating around in the air until photosynthesis came around. So just keep that in the back of your mind, because we want to talk about eukaryotes now. Eukaryotes. So draw a line in your notebook if you need to. Separate this page, because we're going to be talking about a new kind of thing, a eukaryotes. So let's go back to that early version of life. We've got our prokaryote, right? And these prokaryotes are dividing and they're making lots more babies. And some of them, they would realize, well, I can just eat the other prokaryotes that are around there. So they would start to surround those other prokaryotes, bring them into their cells. So they got them inside, and then once they're inside, just like you would chew and swallow things, they'll break them down. And they'll use all of the materials and energies from the prokaryote they swallowed to power their cell. This happens even today. If you look at amoeba, they do this same process. Your white blood cells do this same process. This process is called phagocytosis. Not really the focus of today's video, but I'm just throwing that out there for you. But after these cells would eat them, they'd need to go and find another cell. So they'd go and they'd do this again. And they'd keep doing phagocytosis until one day something different happened. They didn't digest the cell they swallowed. Instead, they kept that cell around to make energy for the big cell. So now we have one cell living inside of another cell. I'm going to give you a big word. I want you to write this down. It's called Endosymbiosis. 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 Now this word sounds scary, it sounds big, but it's not. Let's break it down one part at a time. So let's take this endo. That's where we get our word enter from. It means inside. Okay, endo, inside. Sim, okay, sim means together. Together. Bio, you know that one. That means life. And then this word osis means process. Okay? Now, the interesting thing about Greek words and other Latin words that use them a lot is if you read them, like you take their roots and then you read them backwards, it's going to tell you what they mean. So, the process of life together inside. The process of life together inside. That's telling us what this word means. All we have to do is break it down and think about the individual parts and kind of read it backwards. We have one cell living inside of another cell. What if I told you that your cells actually had bacteria inside of them? Not around them, not on them, but living inside of every single one of your cells is another cell. And that cell is giving you the energy you need for your life. That's a pretty crazy and bold claim, I think, but that is what we think as scientists, as biologists, has happened. We think that one cell got swallowed by another and is now living inside of your cells. We think that this cell evolved into mitochondria. And then we think, of course, these cells would divide and continue on. And we think this same process happened again, but instead of it being a proteobacteria, it was cyanobacteria, bacteria that could do photosynthesis. And that is where we get one cell with two different kinds of bacteria in it. And we think this line eventually became animals and fungi. That's where we came from. And we think this line eventually became plants. It's a pretty crazy and bold claim. So let's look at the cells, right? Let's just look at what an animal cell looks like. Let's look at what a plant cell looks like, and we'll come back to this idea of the endosymbiotic theory. So before we leave it, let's give this thing a name. Endosymbiotic theory. Okay, endosymbiotic theory. And then let's just draw a cell. Oh, no, we're not doing endosymbiosis. We are doing eukaryotes. This is why pencil is great when you're taking notes, in case you make mistakes. All right, so let's draw our eukaryote. This one's going to be a lot bigger than our prokaryote. Okay, much, much larger than our prokaryote. So we'll draw that big old cell. Just draw a big circle. This will be an animal cell. Animal cell. This is like your cells. So these are eukaryotes, which means they have a nucleus. So we'll draw the nucleus there. Just a circle inside of our circle. Inside that circle, there's another nucleus called the nucleolus. I know, kind of looks like a creepy eye now. Okay, so let's label this already. We've got the nucleus. And we have the nucleolus. And of course, we have our cell membrane. That's right, eukaryotes and prokaryotes both have a cell membrane, and this cell membrane is serving the same functions in both. The prokaryotes were using it to regulate what goes in and out of the cell. The eukaryotes are using it to regulate what goes in and out of the cell. Now, eukaryotes also have this other thing surrounding their nucleus, and it's kind of this crazy foldy type of organelle. So it kind of looks like a hand grabbing of something. You just kind of draw back and forth different layers around the nucleus. This is called the endoplasmic reticulum. Or we can just call it the ER. Don't confuse it with the emergency room, though. The endoplasmic reticulum. Okay. Now, the interesting thing about the endoplasmic reticulum is there are two kinds. There's the smooth and the rough. The smooth looks smooth, and the rough looks rough. So on one half, draw little dots. Okay. And these little dots that are in the endoplasmic reticulum are also scattered around the cell. So you can just draw a few little dots around your cell. These dots are ribosomes. Oops. Gotta spell. Ribosomes. Okay. So we've got our cell membrane, we've got the nucleus, nucleolus, endoplasmic reticulum, the smooth endoplasmic reticulum, which does not have ribosomes, and the rough endoplasmic reticulum, which has ribosomes. Okay? And then coming out from this endoplasmic reticulum, there's this like layer of, we could just draw a bunch of ovals, kind of like I am, and it's these layers and layers of membranes. And this is called the Golgi body. The Golgi body is these layers of membranes. And we'll talk about this little area here in a minute. But we also find inside of our cells these little spherical types of organelles called lysosomes. Lysosomes. And we also find, you probably heard about this in middle school, or if you're in middle school right now, you're going to hear about it for the first time. These are mitochondria. Now, mitochondria, you can just draw an oval and then draw kind of like this squiggly line inside of that oval. Mitochondria. Now, you might be wondering, why is that CH making the K sound? Well, that's because in science, most of the time, the CH just says K. That's why you say chemistry and not chemistry. That's why we say mitochondria and not mitochondria. Okay? All right. So we're going to have different lysosomes around the cell, so we can draw a few more circles. And we're going to have a few more mitochondria inside your cell, because you typically don't just have one, you typically have several inside of your cells. Now we've got one more thing I want us to draw, and a lot of people get this one wrong, and they say that plant cells only have these and not animal cells, but it's not true. We have an area inside of our cells that we can use to store things called the vacuole. Vacuole. Okay. Now, in animal cells, they tend to be pretty small, especially compared to plant cells. We'll look at that in a minute. There's one more thing that animal cells have that I want to make sure you know. It's these special kinds of proteins that help the cell divide, and they're called centrioles. Okay, so now we've got our animal cell. This would be a great thing to, like we did earlier, color in while I describe what each part of the cell does. All right, we already talked about the cell membrane, how it regulates what goes in and out of the cell. The nucleus is going to be storing DNA. Okay, now earlier, if you remember, we had our nucleoid region, and they have those similar names, nucleoid, nucleus, for a reason, and that's because they both store DNA. Okay, they both store DNA, and in fact, it's the nucleolus that stores the DNA, and the nucleolus also makes ribosomes. Okay, now in the prokaryotic cell, that's just going to happen in the nucleoid region, but inside the eukaryotic cell, we have a special place for it. Just outside of that, we have the endoplasmic reticulum, or the ER. The smooth ER makes lipids. So we'll write that down here. Smooth makes lipids. Okay, that's like fats and oils. I'm running out of room. I apologize. And then the rough ER, it's got ribosomes in it, so it's going to make proteins. Now, it's not actually the endoplasmic reticulum that's making the proteins. They're just helping out. The endoplasmic reticulum acts like scaffolding. It acts like a structure. So if you've ever seen on the side of a building, they have all those different like layers when they're building the building. They have all that different stuff on the building. That's scaffolding, because it's allowing things to work off of them. It's allowing those workers to work standing up and be more comfortable and make it easier to build the next layer. That's what the endoplasmic reticulum is doing for these proteins. If you remember, proteins are the tools for the cell that they're going to use to do their metabolism, to break things down and to build things for the cell. So this rough endoplasmic reticulum is helping make those proteins, helping make these special tools. So you can almost imagine that the endoplasmic reticulum is like a giant factory. Now the Golgi body is going to be working kind of with the endoplasmic reticulum, because the Golgi body is going to take the things made in the endoplasmic reticulum and it's going to export them outside of the cell. It's going to push them out. This is very important to make like your saliva, your mucus, the oil on your skin. All of that gets exported from the Golgi body. Now we skipped ribosomes, but you remember ribosomes from earlier, they make proteins. Golgi body is going to be exporting the proteins made in the endoplasmic reticulum and the ribosomes. Lysosomes are very important, especially because when we were talking earlier about endosymbiosis, right? We have the cell getting swallowed and then it gets broken down. That cell gets broken down by the lysosomes. The lysosomes break food down. They digest. They digest things inside of the cell. If we've got old proteins, if we have broken machinery inside of our cells, the lysosomes will break that down. If we have cells that have been swallowed by our white blood cells, then the lysosomes are going to break those down. The mitochondria are very, very important, and they provide energy. In fact, you should star mitochondria. They're super important for what they do. The mitochondria provide energy for the cell, and they do that by making ATP. We'll talk about ATP later. Vacuole. These vacuoles, they act like a pantry for the cell. They can store things. Inside your fat cells, your adipose cells, you'll have large vacuoles that will store those fat for later use or for padding. Okay, adipose cells are important parts of our bodies, so don't let that word fat discourage you or anything. The vacuoles are just storing the things for later. And the centrioles, these are these little lines inside of our cells, they're going to help the cell divide. At some point, this cell will want to make a new cell, maybe we get injured, maybe we're still growing. And so the nucleus is going to go away, and you'll see the different chromosomes inside, the different DNA, and the centrioles are going to help that DNA split apart. All right, so that's our animal cell. Here's our animal cell. You can see all the different organelles. And there are some other specialized organelles, but this is just our generic animal cell. All right, we're going to put this aside. If you need to, you can pause the video or come back to it. And we're going to look at the other eukaryotic cell we need to know about. And that, of course, is the plant cell. All right, now the nice thing about plant cells and animal cells is that they actually share a lot of structures. So let's just draw a big rectangle for our plant cell. Big rectangle. This is our cell membrane. And just outside of that rectangle, draw another rectangle. This other rectangle is the cell wall. Notice, animal cells did not have cell walls, but plant cells have cell walls. This is a big difference between these two kinds of cells. All right. Then let's draw the nucleus, because plant cells are eukaryotes, the nucleus, and they'll of course have a nucleolus. Nucleolus and nucleus. They will, just like the animal cells, also have this endoplasmic reticulum. And I'm just going to write ER because we already did that earlier. And I think this video is getting a little long, so we're going to try to keep it short. They have both smooth ERs and rough ERs. And they also have Golgi bodies. And they, of course, also have ribosomes. If you need to, you can go back and pause this video and draw what I'm drawing. But I think it's pretty easy. We've got our ribosome, and they also have lysosomes. And they also have, and this is what's important and unique and different when I was talking about the vacuoles, plant cells have large vacuoles. They have these really large storehouse areas. That's very important for plants. All right, now let me think. Did I miss anything? Oh, yeah. Two really, really important things. Plant cells, like our cells, have mitochondria. So I'm drawing them just a little bit smaller here for this plant cell. But they've got mitochondria just like we have mitochondria. Okay. I'm kind of off the camera, I'm sorry. Mitochondria here. And they also have, what's unique to plant cells from animal cells are chloroplasts. Chloroplast. Okay, so I think all of these other organelles we talked about before, but the chloroplast we haven't. So I want you to star this chloroplast here. Chloroplasts do photosynthesis. Photosynthesis. Okay, they're capturing light energy and using it to help the cell maintain homeostasis, to give it all the power that it needs to maintain the cell. Okay, if you need to, go ahead and pause the video here to draw and to label the rest of your plant cell. But I want to go back to, really quickly, this giant claim I made earlier about endosymbiosis. and how our animal cells and fungi cells as well came from one cell that swallowed another cell. And plant cells came from one cell that swallowed two other cells. So notice we've got mitochondria, and plants have mitochondria, and chloroplasts are also found in plants. So plants have these two organelles. Okay, so let's go to another page. We got lots of notes for this one, right? I'm going to quickly organize this. This is going to be the last thing we talk about, so stay with me. Endosymbiosis, right? We've got our mitochondria, and plants have chloroplasts. Mitochondria and chloroplasts they look like bacteria. They behave like bacteria. Their DNA is circular. They've got their own ribosomes. They have all kinds of things that look specific and unique to them. And that's why we think eukaryotes came later. We think that mitochondria and chloroplasts were once bacteria that lived outside of the cell, were swallowed by another cell, and then became a part of this larger cell. And so when you think about prokaryotes and eukaryotes, I want you also to be thinking about mitochondria and chloroplasts, because I think it will help you understand more holistically what's going on. We've got not only these bacteria that are simple living things, but we have those bacteria inside of our own cells. And having a cell inside of another cell allows for much more complex metabolisms. I want you to think of a one-room hut. Everything that the cell does has to happen in that room. Everything that somebody does who lives in a one-room hut has to happen inside of that room. Now I want you to think about a mansion. In mansions, they have a room for everything. They have a room to sleep in, a room to eat, a room to play bowling, a room to keep their library, play video games, watch movies. They've got a room for it all. And this is the difference between a eukaryote and a prokaryote. Eukaryotes got a nucleus. They were able to compartmentalize what's happening in each of these different rooms. So we've got a different thing going on in the nucleus. It's got its one job, right? The chloroplast is different things going on. We've got a vacuole, different things, storehouses, right? We have all of these specialized organelles, all of these different areas which can have different special tasks. Compare that to our prokaryotic cell. It's got one room. Everything that's happening here has to happen in that one space. So by adding a cell inside of a cell, it's like putting a room inside of a building. We're having a separate space to do things. We're having a unique spot. So we don't have to double in order to have specialized areas to do things. And this is a fundamental and radical change for our cells. This is why prokaryotes are larger than eukaryotes. This is why they're more complex than eukaryotes. It all has to do with one cell living inside of the other cell. It all has to do with their ability to create a nucleus and compartmentalize. This part of the cell is doing one thing, and another part of the cell is doing something different. It goes back to the creation of the nucleus and the development of membrane-bound organelles. That means they've got a membrane. So I know we kind of drew these little circles here, but I'm going to go ahead and fill these in to kind of make this point. Prokaryotes do not have membrane-bound organelles. They don't have any other cell membrane. It's just that one. But eukaryotes have all of these other membrane-bound organelles. Chloroplasts have a membrane. Vacuoles have a membrane. Golgi bodies, lysosomes, they all have membranes. So almost every single one of the organelles I showed you, with the exception of centrioles, that are found in eukaryotes but not prokaryotes, have membranes. All right, so take a moment and in the comments below, tell me what you think. Do you think we actually have prokaryotes living inside of our cells or not? And what do you think about all these different organelles? Which ones do you think are more interesting than the others? Which ones do you think are more useful for the cell? Thank you for watching.